right. I honestly don't remember where we left off. Get that turned on. The R's, right? Malthusian parameter. Instantaneous growth rate. Right, intrinsic rate of growth. So I know we did, let's see here. I got a couple of those. I think it's this one. This one? We can pick it up. Yeah. All right. I think you got everyone. All right. Well, it looked like three B and D relationships. Got it. All right. So it's good to see that some of you survived the Halloween snowpocalypse of 2020. Unfortunately, we lost out on our lab, our Tuesday lab. I sent out an announcement, so Monday I know has the lab, Wednesday, Thursday should have this lab, which means everyone should be on the same page except on Tuesday. So what I did was, here's our Blackboard page. Under the lab, we have demography. All right, so what I've done, I went ahead and made a video on how to create a life table, how to fill out a life table. I actually had two examples. One would be the equivalent of like a male-only table where we don't have a birth schedule. So we just use the information to calculate our survivorship curve and then life expectancy. And then I had the equivalent of a female-only table where you would do survivorship, life expectancy, but also because of the birth schedule, you can calculate your generation times and your net reproductive rates. Okay, and then the third video in this playlist is how to take the survivorship column, the LX column, and make a survivorship curve, but not just a single value, not just a single curve on a graph, but to have two curves on the graph and to make sure that we're actually plotting it on the correct scale, because as you go back to that survivorship curve slide, we said that the y-axis is the log scale. It's on the log scale. All right. So if you missed lab on Tuesday, that doesn't mean that you just don't have anything. We'll still take your quiz next week. I have two quizzes next week. Surprise, surprise. Unfortunately, they're both short. They're both pretty short. But what you're going to do is you're going to need to work through the lab. The data is in that lab handout. So you'll work through the lab, make the life history or the life tables, and then from the life tables, you'll with that practice, you'll be able to do the homework. So on this homework assignment, there's basically 4 graphs that you're going to make. You're making 2 graphs per problem. There's a question 1 and a question 2. The first 2 graphs of each of those problems is survivorship curve. So it's kind of what is in the video. All right, you're going to have a survivorship curve, or one graph that has 2 lines on it, one for a male and one for female, of Texans that died in 1970. All right, and then in question 2, you're going to combine the sexes and do a one survivorship curve for 1970 and one curve for 1870 Texans, so we can compare the 100-year difference in survivorship curves. And then the second graph for each is a mortality curve, and that's our QXs. Same type of thing, but with the QXs, we don't plot on the log scale. So check out YouTube playlist. If you're unsure, let's say on Monday or on Wednesday or Thursday, if you're still a little bit unsure how to make these things at home, watch the videos. It kind of goes through the problems in the handouts. I did update this handout, so some of you on Monday may have noticed it, but I updated it just because an equation wasn't quite right. So if you remember it, I'd give this equation down here, and the one that's interested is this TX. Monday already knows we use this to calculate our life expectancy. This is the sum starting at the age of interest all the way to the end of our table. I had this as a 0. It actually starts at the age of interest and then goes on to the rest of the table. So I talk about that in the handout. The other thing is this allelopathy handout. So this is, I think, where Tuesday loses out. This gives you the introduction. This is actually, I'll count it as a 3-week lab. It's technically a 2-week lab, all right? But we talk about this now because what you have to do is get things set up now, this week, so that in our lab next week, we actually set up our plates to test these allelopathic compounds, all right? So what you're going to do is whoever your partner was when we did the ragweed data, get with them and figure out what we're going to test. So the whole idea of this allelopathy is sometimes the plants produce some compounds that either have a positive or a negative effect. Usually, we're going to be interested in this negative effect. Are plants producing some sort of compound that has a negative effect on other plant species? All right. We're going to test this using grass. We're going to look at germination rates, and we're going to look at rootlet lengths. All right. So what you're going to do is to make what I call leachates, or teas. You're going to pick out 2 plants. All right. I give possible species here that you can pick, and you are going to take the plant material, put it in a container, have water in it, and you're going to let it soak. This is not something you can do the night before. You need to do this at least 3 days ahead of time so that it gives time for the water to suck out the compounds and get it into that water. All right? You don't need a whole lot. I said in the email about 8 ounces. We're going to use maybe 1 ounce, 1.5 ounces on our plates, but 8 ounces is a good one. Some people have come in with bottles. They've jammed leaves or needles or whatever into these bottles, all right? And if the water overflows, it overflows. That's fine. But they've used water bottles, and they've come in with 2 water bottles. I'm going to ask the third one. So if you use this, I don't know, Niagara water bottle, and who knows how long this has been up here, all right, bring in the sample of the water as well. Because what we're going to do is set up plates, 3 different plates. We'll set up a plate with just the water that you used, a plate with one of the plants, water from one of the plants, and then the third plate is a plate with the other plant. All right, that's going to have to be set up. It's up to you, and I'm going to tell my class, remember, we're doing an experiment. So think about all of the factors that can go into it. Don't pick green leaves for one tree and then brown leaves for a second tree. That you've just introduced live versus dead in addition to testing the species. All right? If you pick green leaves for the second. You can pick sticks. You can pick the nuts. It's up to you. This is kind of open type of experiment. So you're going to make these leachates. You're going to bring them to lab. If you don't have them, then you're going to fail that lab because we can't set up the plates. Get this set up at least 3 days ahead of time. The second thing that you're going to do is to write up a justification and a hypothesis. This is going to tell me why you chose those plants. Why did you choose them? What did you see? And all I can say is just think about the plants, do a little bit of research on them, think about it. Do they produce any sort of chemicals? All right, do they know any sort of chemicals? Maybe they're used, maybe the tree product or the plant product is used in some sort of manufacturing process. Maybe that has maybe it's a chemical aspect that we're using. Maybe it's an observation. Go out and look at the trees. Make an observation where it might lead you to the possibility that one of these trees, but not another, would produce an allelopathic compound. All right? You're going to write a paragraph that kind of describes what tree you're picking, or what trees or shrubs you're picking, and justifying why. Why are you doing that? Why are you interested in it? And hypothesize what you might see. All right? It's pretty uninteresting to pick two plants and say, yeah, I don't think these are going to have allelopathic compounds. Try to pick at least one where you think you'll have an allelopathic compound and maybe the other is not, and you're going to use that possibly as another control. All right? This justification is one per group. So contact your lab partner, your partner that you worked the ragweed lab with, narrow down what two tree or plant species, what type of plant material you'll use to make this leachate, and then sit down and write a paragraph. This is one paragraph, and this is probably on the longer side of things, but this is about the size. Right? I don't need a 2-page justification. Just something that kind of tells me that you've thought about it. All right? So I described the leachates. I say it holds a minimum of 12 ounces, but again, we only really need about 8 ounces. So, one of these water bottles will work. I've seen even the really short, stubby water bottles used. That can work. I would say, based on experience, you should probably try to have as much plant material as possible to try to maximize the likelihood that we have a high enough concentration of the chemicals. So if there is an allelopathic effect, we can detect it. That's about what I can say about it. All right? So I normally talk about this in my labs, and I'm sure Dr. Skipper talked about it yesterday, Monday. So now everyone hears it. This is the spiel. Maybe we'll save some time in our lab. All right, questions? Any questions over lab or anything else? I am glad classes weren't canceled today. But if they were, we probably just would have canceled the lecture. No video. All right. Growth equations. We talked about discrete generations, right? Discrete generations produce a growth curve like this pattern, the sawtooth pattern. Why does it produce this pattern? Because in these discrete generations, we're going to have birth pulses. All right? And the birth pulses will cause this rapid increase over a very short amount of time. All right? That causes our spike. And then between our birth pulses, we will have natural mortality. What type of natural mortality are we talking about? Accidents. The animal runs out into the road and gets hit by a car. We're talking about predation. We're talking about disease. We're talking about parasitism. All of those things I'm classifying as natural mortality. All right? So you have natural mortality that causes population size to decline, and then you hit your birth pulse. All right? Our net reproductive rate actually measures the distance between our peaks or the distance between our valleys. All right? Either one is going to work. Now, as we move from discrete to continuous generations, what we're doing is reducing the amount of time here between our birth pulses. And we're reducing it so much that eventually we get a smooth line. We're reducing those saw teeth down until they're basically really fine saw teeth and then a smooth type of pattern. And this is our continuous generation growth curve. This is an exponential growth curve. All right? So this growth curve is smooth. And trying to estimate changes in population size could be a little bit confusing. However, if you recognize that the change in our population size is actually the slope of this curved line, then things become a little bit easier. All right? The equation becomes a little bit easier. So this slope, at this point, we can kind of think of our slope as being a straight line, and this is our tangent line. All right? And this slope tells us the change in the population size or the population growth rate. Now that growth rate, if we move along our line, is going to change. It starts out shallow down here, and then as we increase in its size, we're going to have a very steep line. So our growth rate's going to vary based on our size, and it's also going to vary based on some term, some fixed population growth factor. And that growth factor is R, our instantaneous growth rate, the intrinsic rate of growth, or also called the Malthusian parameter. All right, so this R is the potential growth rate. This is just based on our population. This is, you can think of it as kind of like an average number of individuals that are produced, but this scales from 0 to 1 because what it's talking about is that if R is 0, then our individual doesn't really have any more potential to add or take away from our population. It's just replacing individuals there. All right? So our relationships here, unlike lambda and R naught, is that our intrinsic rate of growth, if it's greater than 0, then we have a positive population growth rate. We have growth. If it's below 0, we're in the negative, so we're going to see decline. And if it's equal to 0, we'll have no change. All right, so I think this is where we left off. All right, so can we predict population size? Can we predict population growth rates? Yeah, we can. We can do both with this exponential growth curve. All right, so our population growth rate, the slope of that line, that dn over dt, is equal to our intrinsic rate of growth times n. And this is where you can see that our population growth rate depends on our population size. And it should make sense. The more individuals that are there, the faster our population is going to grow. Even if each individual only produces 2 offspring. Right? If you have 100 individuals and each of them produces 2 offspring, well, you have 200 individuals coming into our population. That's a change of, let's say, 200. Let's be simple and say it's an increase of 200. But if you have 1,000 individuals and each one produces 2, now we're adding 2,000 individuals. So this is where our population growth rate changes with our size. It's this equation here. What about predicting population size after a fixed amount of time or after a number of generations? Well, that is this equation here. We're predicting our population size at time t, and that's going to depend on wherever we start, our starting population size times e raised to the RT. So e is our exponent. This is our exponential term. On the calculators, mine is on the ln button. So I have to use my shift key and then the ln to get to that. All right. In Excel, it was that exp function, where we did equals and exp. And R is our intrinsic rate of growth, t is our time. So this equation predicts our population size after t time. Could be hours, could be days. It's going to be basically whatever this R units are. So if R is in cells per hour, then t is in hours. If R is in individuals per year, then t is in years. All right? So here's an example of how we would use this equation. So yeast exhibit continuous growth. All right? Their generations are, you can't tell one cohort from another with yeast. All right? And yeast are a classic example. Bacteria are also a classic example of this exponential growth. So what we'll say is yeast exhibit exponential growth, and their intrinsic rate of growth is 0.3 cells per hour. So number one, is this population going to increase or decrease? It's what? It's increasing. Why did you say that? Because R is greater than 0. Okay, so our population is going to increase at this point. So we know it's going to increase. So first part, if we start with 10 cells, then how many will we have in 10 hours? Note, my R was cells per hour, so my time is going to be in hours. So as with anything, what I like to do is go ahead and write out all the information that's given to us in our problem. So let's start with what do we know? What is given to us? So we have R. Let me move this. I guess I'll just hold this. R is 0.3 cells per hour. All right, that's given to us in the problem. And for part A, we're saying our starting population size is 10 cells, and we're asking how many will we have after 10 hours. All right, so we have our N0, our starting population size, and we have our time, 10 hours. And I ask how many individuals, how many cells do we have after this 10 hours? So we're asking basically that to solve, figure out what this n sub 10 is. So our equations, we have two to choose from. We have that equation, dn over dt equals r times n. Are we going to use this one? What's that? No. And why not? Because this equation calculates our population growth rate. This calculates our change in our population size. That's not what we're asking. We're asking for an actual population size. So we're going to use our next equation, which is this. N sub t equals n naught times e to the rt. I have n sub 0, I have r, and I have t. E is built into the calculator. So I'm just going to go ahead and substitute all of our terms. So we start with 10 cells, e raised to the r, 0.3 times our t. And then we'll use our calculator. I've been unsuccessful in capturing this calculator screen. So here's our calculator. We start off with 10 times E. On the Windows calculator, it's this EXP. Should be. I'm in. How come EXP hasn't appeared? Oh, there it is, E. E raised to the parentheses 0.3 times 10. So hit equals, and I get a population size of 200.9. Or I'll just say 201. That's our population size. Make sense? All right. Second question. When our population is 5,000 cells, then how fast is our population increasing? That is using this equation. How fast something is increasing or how fast something is decreasing, that's our change. All right? So for part B, we still have R, it's given to us in the problem, but now we list our population size of 5,000. Right? So if we have 5,000 cells, then what or how fast is our population increasing? So we're going to use this dn over dt. This is r times n. So it's 0.3 times 5,000. And what does that give us? Pull out my calculator. 0.3 times 5,000. I get 1,500. And if I use my units, this is cells per hour because that is the units of our R. Questions? Plenty of examples out there. Plenty of examples out there. And I will have some, and I'll show you those when we finish up this lecture. All right. So there's one more thing that we can talk about, and it's doubling time. So doubling time is the time required for a population to double in size. And this is very important when we're trying to raise populations for either the food industry, the pharmaceutical industry, or perhaps even if we're interested in food production. All right? Why is that? Because sometimes we need to grow up these microorganisms to a certain level that then become usable to us. And we need to know how long is it going to take to get to certain points. And this is where doubling time comes in. So doubling time is t, and it's equal to the natural log of 2 divided by r. And I think this is kind of important to recognize that this equation doesn't include our population size at all. Doubling time only depends on the intrinsic rate of growth, r. That's all that it depends on. All right, so if we compare two different strains of, let's say, bacteria, the bacteria with the larger R is going to have your shorter doubling time. Right. If you have two strains of yeast and they have the exact same R, they're going to have the exact same doubling time. It doesn't matter what their population sizes are, doubling time only depends on our intrinsic rate of growth. So here's our example. We have two strains of yeast. Strain 1 exhibits an intrinsic rate of growth of 0.2, while strain 2 exhibits intrinsic rate of growth of 0.3. All right, so calculate the doubling time for each strain. We have our equation, all right, natural log of 2 divided by r, so go ahead and do that. We'll check your answers. I'll write it out. That's still on here. All right, so hopefully you have an answer. Strain 1, 0.2, that's our R. We asked for the doubling time. Doubling time, we have simple equation. So, pull out my calculator. It's the natural log of 2. 2 natural log divided by 0.2. And I get a doubling time of 3.466. We'll say 3.5. Let's say this is cells per hour. Let's say R is in cells per hour, so that means our doubling time is 3.5 hours. All right. Strain 2. Go out to my calculator. 2 natural log. Not every calculator is like that, by the way. So my calculator, I can hit ln and then the 2. But some of them, you have to hit the number and then ln. I guess it kind of depends on what brand you have. Anyways, it's natural log of 2 divided by 0.3. We'll say 2.3 hours. All right, so strain 2 has the shorter doubling time. Does that make sense? Yeah, it should. Has the larger R, has a higher reproductive potential. All right, it's going to replicate faster than this one, which means it takes a shorter amount of time to double its population size. So the question 2 then, or part part B of this, is if strain 1 starts with 1,000 cells and strain 2 starts with 250 cells, then which strain will have the fastest doubling time? What's your answer? Which one? Strain 2. Why'd you say strain 2? Yeah, it doesn't have anything to do with population size. This information was there just to screw you up, just to throw you off. You're going to look at this and say, oh my goodness, Dr. N gave me population size. I have to include it in my problem. No, you don't. You don't. This was here just to drive home this point. Doubling time only depends on the intrinsic rate of growth. That's it. Doesn't matter how many individuals we have. It only depends on R. So that those with the higher R, with the higher reproductive potential, will have the shortest doubling time. All right? Questions? All right. Exponential growth. How likely is it? It's not super likely. All right? But it's possible, and we see it. All right? Why is that? It's because if we think about the exponential growth curve, our curve is smooth, all right? It goes up, and it would go up to infinity, which means that our population could grow unchecked. How likely is that? How likely is it? What do you say? How likely? Checked. What's that? No, that our population size will grow to infinity. Not very likely. Not very likely. Why is that? It's because we have processes in our population that regulate it. Things like resources. Resources aren't unlimited. You know, the more individuals that are there, the more the resources get consumed. That's going to cause a slowdown of our population growth rate. You're going to have space. You can only fit so many bacteria in a single agar plate. All right? You can't go beyond that. All right? So our exponential growth has these 5 assumptions that must be met in order for the population to actually exhibit this exponential growth. So what are they? What are these 5 assumptions? So assumption number 1 is that we have a constant R, a constant intrinsic rate of growth, a constant instantaneous rate of increase. All right? R can't change. It has to be fixed. Now you could say, okay, yeah, R is always going to be the same. We calculate it at 0.2 or 0.3. But what I want you to consider is that R can change based on the surrounding conditions. So individuals that have plenty of nutrients and plenty of space have a higher reproductive potential, usually, than those populations that don't have that same stuff. So we can't have any sort of limiting resources present. We can't have any feedback mechanisms at work that would alter this intrinsic rate of growth. All right, and this is a big one. We need this. If we don't have this constant R, then we're not going to see exponential growth. The second assumption is that we're working with a closed population. No migration. All right? And we've already kind of made that assumption. We've said as we introduce these equations, we assume net migration is 0. It makes the equations easier to work with. We don't have to deal with immigration or emigration. Is it realistic? Sometimes. I mean, you think about yeast in a nutrient flask. Yeah, that's closed population. You don't have wild yeast coming in, or you shouldn't have wild yeast coming in. So no migration. In the natural world, probably not, but in a lab setting, yeah, we can make this work. The next 2 assumptions, we have no genetic structure and no age or size structure. Both of these are tied to R. All right, if we think about genetic structure, we can't have a population where we have different genetic strains that we wouldn't know about unless we sequence the populations. But perhaps those strains confer advantages and disadvantages in terms of reproductive potential. We can't have that because we need all individuals to basically have the same R value. All right? This is kind of the basis of natural selection. individuals that are best adapted for the environment are going to have the higher R value. They're going to leave more offspring. All right? So we can't really have it. We can't have that genetic structure. Likewise, we can't have the age or size structure. We can't have larger individuals having a higher R value just because of their size. All right? Not possible. We can't have older individuals having higher R values or lower R values just because of their age. Again, we assume that this R applies to all individuals across our population. And our last assumption is that we can't have time lags between mating and birthing events. All right? We are modeling an instantaneous change. And just to kind of demonstrate that, here's our curve. This assumes that when reproduction happens, our offspring enter that population at that time. If we didn't have that, then we could have a curve that looks more like this, where we have our mating event, and then all of a sudden, you'll have an introduction into our population, and you'd get these kind of jagged type of curves. So with this time lag, we can't have the time lag. Once we mate, our offspring are going to be produced almost immediately. And that kind of goes to what R actually means. You know, the instantaneous growth rate, the intrinsic rate of growth. We're modeling instantaneous change. We're not modeling time lag changes. All right, so if all of these assumptions are met, then we'll have exponential growth. When does this happen? Usually it happens when we're at a very small population size. At those small population sizes, they're going to have minimal feedback mechanisms. Populations are going to see these unlimited resources, and usually, since you have such a small size, they're not sending individuals out as migrants. They're not really attracting any migrants themselves. All right, so summary of this. Populations change over time. Right? We know that. We knew that. All right? This populations change over time because of different processes, but the main ones are births and deaths, and immigration and emigration. Births and immigration add individuals. Death and emigration removes individuals from our population. And our population size in our next generation depends on these four processes, but also depends on what our current population size is. What our population size today helps us make predictions about population size tomorrow. Now, even though we assumed migration is zero, assume this part is zero, all right, dispersal does play an important role for populations. In terms of the population itself, it's going to affect the size of the population, and that's through its effect on immigration and emigration. But it also affects our genetic similarity, our genetic variation of our different populations. And say, go back and look at the FST values again. Look at how migration and gene flow affects this value, because this value summarizes or concisely describes genetic similarity between populations. We then introduced our mathematical population growth models, and we broke it up between discrete generations and continuous generations. So if we're working with discrete generations, then we're using one of these equations, and the equations are basically the same. The format's the same. Our population size at t time is equal to our growth parameter raised to the t times our starting population size. The growth parameter is the one that changes. So if we have non-overlapping generations, we're using lambda, our finite rate of increase. If we have overlapping generations, you're going to use R naught, the net reproductive rate. What's the difference between these two? Mathematically, there is no difference. They function basically the same in these equations. But conceptually, lambda only deals with births. Our net reproductive rate accounts for both births and deaths. When we work with a continuous generation, what we've done is taken that time between birth pulses and made it basically infinitesimally small. Super, super, super tiny. So our growth curve looks very smooth. All right? The slope of that line, of that smooth exponential growth line, tells us the change in our population size, the population growth rate. That's dn over dt. And for an exponential population, for a population growing exponentially, we calculate that by multiplying our net reproductive, our intrinsic rate of growth by our population size. And I didn't give n0, I just gave n because our population size, the growth, the change in population size varies based on our population size. If we want to predict our future population sizes, then we use this equation. So we use our original population size, n sub 0, and we multiply by e raised to the rt, where e is this exponent, this exponential term. All right, it's on our calculator. We use that. And then r is our intrinsic rate of growth. T is our time. You can think of this e to the rt as kind of like net reproductive rate. And I think I actually do that later on. I show you how you can convert between them. And then the last equation that we introduce is doubling time. Why is doubling time important? You know, if you're in the food industry, if you are looking at maximizing production of, let's say, game animals, we want to try to maintain populations at a certain size, or maybe we need to wait a certain amount of time before we start allowing public to come onto the lands, start harvesting. How much time does it take? This is where the doubling time comes into play. It only depends on R. All right. Good, I think we buffered. So, one last thing. If you go up to lectures, the course content, I have problem sets. And the first problem set is population growth, and this went live. All right, I've got actually 3 things. These are practice problems, population growth practice problems. Problems that, just like what we've worked with here, all right? These are to give you extra practice to work through them. And there's a YouTube playlist here that kind of walks you through solving them. So if you try to solve them yourself, if you get stuck and not sure where to go or how to continue, check the playlist. I actually step it out as if I was up at a board. All right? Once you feel comfortable with this, then address homework number 1. 5 questions. It's using all the equations that we covered in this presentation. Work through this. All right? You're not turning this in. What you're doing is taking this quiz, listed as a homework. All right? You have 5 minutes. That's it. That's more than enough time to input your answers, because that's what it is. It's just fill in the blank. You give me the answer. So if I ask, what's the population size in 10 days? You round to the whole number. That's the answer that you put into the quiz. I have this quiz as being capped at 3 attempts. So you can't just go in there and start guessing. Try to do it. Try to do these problems. Have the answers for them. Go in, take the quiz, and I think I've properly set it up so it'll tell you which ones you got wrong. All right, so if you get 4 out of 5, hey, go back. You'll know which one you got wrong. Go back, try it again, watch the videos, see if you can figure out where you went wrong. I'm going to guess that some of the wrong answers will be due to rounding. I just have a hunch that that's going to happen. I say round to the whole number, all right? And I mean that. If it ends with a 5, or you round up. If it doesn't, if it's below 5, you leave it the same. That's going to be our rounding rule. So these are practice problems. You need to work through it, and you need to complete it. Due date, I don't know what my due date is set on here. Let's see. Due date is Friday at the end of the day. All right, you need to work through it, because if you have questions, we can answer the questions in class on Friday morning. Bring them, Bring them to class. Second thing, population growth quiz just went live during our lecture today. All right, it's 12 minutes. It does not have any of these calculations on it where you have to actually calculate population size. That's what the homework is for. All right, and we'll continue that with our next presentation, because now what we'll do is start introducing regulatory mechanisms. Violating the assumptions of exponential growth. This is also due Friday at the end of the day. Gives you time to review, work through these problems, and get them done. If you have questions, please email me or bring them to class on Friday. All right? I'll see some of you in lab. The rest of you, I guess I'll see you on Friday. All right, have a good one.